Here is where we should have finished up, I believe, on Thursday with the notes talking about, body cells are produced by mitosis. Okay? Gametes are produced by meiosis. And we talked about the haploid and the diploid. Remember that the diploid was 2N, the haploid was N. So for human beings, the diploid is 46. We have 23 pair of chromosomes, 46 total. Okay, so the haploid number for humans is 23. The diploid is double that, 46. So basically, a haploid number is 1/2 of the diploid number. Something to remember. Okay? Mitosis produces identical diploid cells. Meiosis, as we'll see later on this week, will produce the gametes, which are haploid cells. So we're going to get into the first part of this meiosis today, and that is stage 1, meiosis 1. Now, what should you be able to do by the time we're done today? Well, you should be able to identify the stages of meiosis 1. Okay, so if you see a picture of them, you should be able to say, okay, yeah, that is this stage. You should be able to tell some key points of what is happening in the stages of meiosis 1. So those are the two things you should get. Our objectives, to be able to identify the stages, and we'll draw these in that pictures that you have there. And finally, to be able to state some key concepts that are happening during the specific stages. Now, I wanted to show you first, before we start talking about these stages, I wanted to show you some different chromosome numbers, because we just said that human beings have how many chromosomes? Okay, what's our number? What is it, Sydney? 46 chromosomes, okay? Well, here are some different ones. The pea plant has 14 chromosomes. A sunflower will have 34. A cat has 38 chromosomes. A pufferfish, 42. We stated ours at 46. A dog at 78. Now, if you were going to state, are those numbers diploid or haploid? Are those the diploid numbers or the haploid numbers? Those are diploid numbers. So what would the haploid of the pea plant be? What would haploid be? What is it? 7, right? It is 1/2 of the diploid. Remember, diploid is 2N, haploid is N. So the pea plant, the haploid, the gametes would be 7. What about the cat? What's half of 38? 19, right? So the haploid number for the cat would be 19. What would the haploid number for the pewter or the puffer fish be? 21, half of 42. Okay, so can you get the haploid number if given the diploid? And vice versa? Given the haploid, get the diploid. Given the diploid, get the haploid. Okay, you're going to see that again. You're going to see that part again. All right. Now, let's get into the stages of meiosis 1. Meiosis 1 reduces the chromosome number by half through the separation of homologous chromosomes. Now, turn to the previous page in your notes. Okay? You should have a picture of what homologous chromosomes are, correct? You just drew that picture the other day. So the homologous chromosomes should be, if you colored them like I had, the purple one and the green one. Those are the homologous chromosomes. Remember the X's. The X's. So, during meiosis 1, we are separating the homologous chromosomes. Interphase. Okay? Interphase precedes meiosis 1. Interphase precedes meiosis 1. So if you remember an interphase, what are the parts of interphase that are important? Think back. Think back to what we talked in mitosis. What are the parts of interphase? G1, S, G2. Gap 1, synthesis, gap 2. So what's happening during interphase? Once again, the chromosomes replicate and the chromosomes condense. Okay? Same thing is happening. Only during meiosis 1, we're going to have during the whole process of meiosis, it's going to be a little bit different than what mitosis was. Now, one thing that is the same, the stages. Okay? How did I tell you to remember the stages of mitosis? Lewis? PMAT. It's going to be the same PMAT. It's just we're going to have that twice. Okay? We're going to have PMAT twice. So if you look here, once again, I just showed a picture. We've seen this before. This is probably the onion root tip. I see the little squares with the cell wall, so I know these are plant cells. Okay? I see a nucleolus here. I see the nuclear envelope. This is an interphase one here. This is an interphase one. Now, I told you that the phases were the same. We just go through them twice. So this is prophase 1. What is happening during prophase 1? Homologous chromosomes pair up. Each chromosome consists of 2 chromatids. The nuclear envelope breaks down. Remember, during that prophase 1, the nuclear envelope is disintegrating. And, ladies and gentlemen, crossing over occurs between homologous chromosomes, exchanging genetic information. So, if you look, flip back once again, we can see the homologous chromosomes, okay? We can see the homologous chromosomes, the homologs. We can see the sister chromatid, okay? The sister chromatid. Each chromosome consists of 2 chromatid. So if I have that X right there that we're looking at, the purple X would be 1 chromatid that has been duplicated. Okay? Now, this whole crossing over deal, the last bullet there, crossing over, this is one way that we get some genetic variation. We'll talk about genetic variation later on in the week, but crossing over is one way. And it's important to note that crossing over only occurs in what phase? Prophase 1. Put that in your mind, okay? Solidify that. Crossing over occurs during prophase 1. That will be a test question. You can bet. Now, what is this whole thing, this whole deal? All right? See here, we've got homologous chromosomes. Let's get the definitions down here. Here is a chromatid right here. Here's a chromatid. Here is another one that is duplicated, so we have the X. See how these X's right here are very close together, and they have kind of crisscrossed right there. What happens in crossing over is that they switch the chromosomes, so to speak. They switch the chromosomes, so this blue X might just have a little green spot down at the bottom. But we'll talk about crossing over later on. What's more important right now is that in the very first circle on your diagram, you are drawing in and labeling prophase number 1. Okay, so after prophase 1 comes metaphase 1. Well, what did we say with metaphase when we were talking about mitosis? Metaphase equals middle, right? Metaphase equals middle. So, the chromosomes, centromeres are attaching to the spindle fibers. The homologous chromosomes line up at the equator on opposite sides. So, this is different. You're going to see here. This is different than what we had occurring in mitosis, okay? Because the homologous chromosomes line up at the equator. Independent assortment occurs. The homologs can line up on either side of the equator. Here is our diagram of metaphase 1. So go ahead and draw that, and then we will talk about it a little bit here. Now let's discuss a little bit about what's going on here. You can see these little red things. These are the spindle fibers. Okay, the centrioles have gone to the poles. The spindle fibers will attach to the centromeres. Now look right here. Look at this blue X. Remember me telling you that that blue X might have a little green tip on it? So what has occurred in prophase 1 there? What do we call it? We just talked about it. Crossing over. Okay? So see how those little parts have switched right there. Same thing with right here. We have crossing over. Those little parts have switched. Now, if you remember back to mitosis, when we had metaphase, all of the little X's lined up. In this one, it is different because they pair up as homologs. So the same size X's pair up together and line up in the middle. So this is where it's a little bit different. The X's line up. If you would remember, and this is going to be just a generic drawing, when we had mitosis, we had those X's lined up as so, being attached with the spindle fibers, as so. Does that look like this? Is that the way it's happening there? No, that is one of the differences that is happening, that the homologous pairs, okay, the homologs are pairing up at the equator. After metaphase comes anaphase 1. Anaphase 1, the homologous chromosomes are separating and moving to the poles. The homologous chromosomes are separating and they are moving to the poles. So, we have this occurring. So go ahead and draw this as the next section of meiosis 1, anaphase 1. Now looking at this, okay, the homologs, the X's are being split. They are being pulled to the poles. In mitosis, if we were to think about mitosis, once again, this is going to be a pretty generic one, but if you remember on mitosis, we were getting something like this happening with the spindle fibers pulling it as so. Is this how that looks? So once again, the homologous chromosomes are separated in anaphase 1, okay? Not the specific sister chromatid. The homologous chromosomes. The final stage of meiosis 1 is going to be what? What's the T? Telophase. Telophase 1, and here we go. In telophase 1, excuse me, the spindle fibers break down. The chromosomes uncoil and form two nuclei. The cell divides, each cell containing half a set of chromosomes, but each still carrying their sister. So the sister chromatids are still together, okay? But the homologous chromosomes have split apart. So the X's of the same size have split apart, but the sister chromatid are still together. And eventually, you know, this is probably one of the early telophase, because eventually this will split apart and we will have two separate cells. We'll have two separate cells. So go ahead and get this guy drawn in your book. So, just a very quick review. If you were given the diagrams, could you pick out which stage of meiosis 1 that you had? Hopefully, since we drew these into our interactive notebooks, we could. Okay? Could you tell me key points? Okay, key points. Could you tell me where crossing over occurs at? Okay, you should, and that is where? Prophase 1. Okay? Can you tell me where the homologous chromosomes line up at the equator at? Metaphase 1. Okay? Where do the homologs start to separate? Anaphase 1. Okay? Where does the two new cells start to form at? Telophase 1. Okay? So those are some key parts to meiosis 1 that you should be able to tell me, and you should be able to go over and identify if need be.